DNA editing, enabling human survival in extreme environments. Introduction. As advancements in gene editing technologies continue at a rapid pace, scientists are exploring the potential for humanity to adapt to extreme environments such as high altitudes, underwater habitats, or even low oxygen planets. This report delves into the implications of DNA editing for human survival beyond Earth, highlighting key advancements, potential applications, and ethical considerations surrounding these technologies. Key applications of gene editing. 1. Adaptation to high altitudes. Gene editing can enhance human tolerance to hypoxia, a condition where oxygen levels are low. By modifying genes associated with oxygen transport and metabolism, researchers aim to improve the body's efficiency in utilizing available oxygen, thereby aiding survival in high-altitude environments. 2. Underwater habitats. Living underwater presents its own unique challenges, including pressure and the need for efficient oxygen utilization. Genetic modifications could potentially enhance human physiology to cope with these pressures, perhaps by improving the efficiency of hemoglobin or developing new pathways for oxygen absorption. 3. Low oxygen planets. For future interplanetary colonization, gene editing could enable humans to thrive on planets with thin atmospheres. This could involve inserting genes that enable enhanced oxygen utilization or even altering metabolic pathways to function efficiently in low oxygen conditions. Enhancing biological resilience. 1. Radiation resistance. Space travel exposes humans to higher levels of cosmic radiation, which can damage DNA and increase cancer risk. Scientists propose using gene editing techniques to incorporate genes from radiation-resistant organisms such as tardigrades into human DNA. Preliminary studies have shown that human cells can exhibit increased resistance to X-ray radiation when these genes are introduced. 2. Detoxification pathways. Gene editing could enhance the liver's detoxification capabilities, allowing humans to better tolerate toxic substances such as heavy metals or acidic environments. This is particularly relevant for potential colonization of planets with harsh environmental conditions. 3. Thermal tolerance. Genetic modifications may also improve the body's ability to withstand extreme temperatures by altering genes related to heat shock proteins and metabolic efficiency. This adaptation would be crucial for surviving in environments with extreme temperature fluctuations. 4. Photosynthesis-like capabilities. A more speculative but fascinating possibility is the introduction of photosynthesis-like capabilities into human physiology, allowing humans to produce energy from sunlight. This could significantly reduce the need for traditional food sources in resource-scarce environments. Gene transfer and experimental insights. Research into extremophiles like tardigrades has provided insights into potential gene transfers that could enhance human resilience. Tardigrades exhibit remarkable survival traits, including the ability to withstand extreme radiation, dehydration, and temperature. By studying their genomes, scientists have identified key genes that could be beneficial if integrated into human DNA. Experimental evidence. Experiments have demonstrated that human cells with inserted tardigrade genes show increased resilience to radiation, suggesting a viable pathway for enhancing human survivability in space. Ethical and policy considerations. While the potential of gene editing is immense, ethical considerations are paramount. The ability to alter human DNA raises questions about the consequences of such modifications, including long-term effects on the human genome and potential unforeseen complications. 1. Global divergence in regulations. Different countries have varying policies regarding gene editing. Nations like Germany and Canada have been cautious, while other countries such as China have shown a willingness to push the boundaries, evidenced by the controversial work of scientist He Jiankui in creating genetically edited embryos. 2. Public perception and acceptance. The societal implications of altering the human genome are profound. There are concerns about the potential for designer babies and the ethical ramifications of enhancing human capabilities. Public discourse and regulatory frameworks will need to evolve alongside scientific advancements to address these challenges. 3. Future research and development. Continued research is necessary to better understand the implications of gene editing. Scientists advocate for a collaborative approach involving ethicists, policymakers, and the public to navigate the complexities of these technologies responsibly. Conclusion. Gene editing holds the promise of enabling human survival in extreme environments, whether on Earth or in outer space. As we stand on the brink of potentially transformative advancements, it is essential to balance scientific progress with ethical considerations. The future of humanity may depend on our ability to adapt. And gene editing could be a key tool in that endeavor. However, as we explore these possibilities, careful consideration of the implications and a commitment to ethical standards will be crucial in shaping a future where humanity can thrive in diverse and challenging environments. The discussion around DNA editing and its potential to enable humans to thrive in extreme environments, such as high altitudes, underwater habitats, or low oxygen planets, reveals several counterintuitive insights. 1. Enhanced resilience against environmental harshness. The idea that humans could be genetically engineered to tolerate conditions typically seen as life-threatening is counterintuitive. Traditional understanding of human limitations suggests that we are inherently fragile in extreme environments. However, advancements in gene editing such as CRISPR-Cas9 may allow for modifications that enhance our physiological resilience. Challenging the notion of human vulnerability. 2. Interdisciplinary genetic transfers. The potential for inserting genes from organisms such as tardigrades, known for their extreme resilience, into the human genome raises questions about the boundaries of genetic engineering. This contrasts sharply with historical views that separate human genetics from those of other organisms, suggesting not only a blurring of species lines but also a radical rethinking of what it means to be human. 3. Self-sustaining energy production. The notion that humans could potentially develop photosynthesis-like capabilities is highly counterintuitive. Humans relying on sunlight for energy similar to plants fundamentally alters our understanding of human biology and energy needs. This could lead to a future where food scarcity is mitigated by biological adaptations. A radical departure from current reliance on external food sources. For ethics versus scientific advancement, the speed of advancements in gene editing technologies often outpaces ethical discussions surrounding their use. While many view these technologies as beneficial for survival in extreme conditions, there remains a significant concern about the long-term implications of altering the human genome. The tension between the potential for human enhancement and ethical considerations about playing God or creating a genetically modified human species presents a paradox that society must navigate. 5. International regulatory disparities. The varying regulatory frameworks across countries regarding gene editing technologies, where some nations are more permissive while others remain highly cautious, suggests a future where scientific progress may not be uniformly distributed. This could lead to geopolitical disparities in capabilities for space exploration and survival, with some nations potentially advancing faster due to fewer restrictions. Challenging the notion of a collective human endeavor in space. 6. Personalized medicine and genetic modification. The concept of personalized medicine evolving into the realm of genetic modification for astronauts raises questions about our understanding of health. Tailoring genetic modifications to individual needs based on genetic makeup might seem like a futuristic ideal but is becoming increasingly plausible. This challenges conventional healthcare paradigms, which generally focus on standardized treatments. 7. Rethinking the human experience. The implications of becoming an interplanetary species through genetic modification fundamentally challenge our understanding of human evolution and adaptation. The idea that we could engineer ourselves to thrive in environments that are currently inhospitable forces a reconsideration of what it means to be human, shifting from a species rooted in specific environmental adaptations to one that can be biochemically altered to fit diverse conditions. These insights not only reflect the potential of gene editing technologies but also highlight the complex interplay between science ethics and the future of human adaptation in the face of extreme environments. The challenges and opportunities presented by these advancements suggest that the path forward may not be as straightforward as it seems. The report on DNA editing and its potential to enable humans to adapt to extreme environments, such as high altitudes, underwater habitats, and low oxygen planets, contains several key insights that may be overlooked by many readers. These insights pertain not only to the scientific possibilities but also to the broader implications and ethical considerations that accompany such advancements in gene editing technology. 1. Potential for enhanced human resilience. One of the most significant yet often overlooked aspects of this report is the potential for gene editing to enhance human resilience in extreme environments. While many may focus on the immediate applications of gene editing, such as enhancing tolerance to radiation, extreme temperatures, or low oxygen levels, the broader implications of these enhancements on human biology and society are profound. For example, the ability to modify genes related to detoxification pathways could allow humans to survive in environments that are currently inhospitable due to pollution or toxic substances. This could lead to the development of new ecosystems where humans can thrive under conditions that are now deemed perilous. 2. Cross-species genetic transfer. The report discusses the possibility of transferring genes from organisms like tardigrades, which are known for their resilience to extreme conditions. This concept of cross-species genetic transfer raises significant questions about the boundaries of genetic engineering. Many people might overlook how this blurs the lines between species, potentially leading to new forms of life that possess traits from multiple organisms. This could have unforeseen ecological consequences, altering existing ecosystems and impacting biodiversity. The significance of this lies not just in the scientific achievement but in its potential to disrupt natural balances and the ethical dilemmas that arise from creating new life forms. 3. Societal and ethical implications. While the report mentions the ethical concerns surrounding gene editing, the depth of these implications is often underappreciated. The global disparity in regulatory approaches to gene editing, highlighted by the contrasting policies in countries like Germany, Canada, and China, suggests that the future of this technology may not be uniformly beneficial or ethical. The potential for a gene editing race among nations where some countries advance rapidly while others impose strict limitations could lead to significant inequalities in access to these technologies. This may create a divide not only between nations but also within societies, where only the privileged could access enhancements that could improve quality of life or survival in extreme conditions. 4. Long-term evolutionary considerations. Another crucial point that may be overlooked is the long-term evolutionary consequences of human genetic modification. If humans begin to significantly alter their genomes for adaptation to extreme environments, what does this mean for the future of human evolution? The report suggests a future where gene editing allows for greater adaptability, but this could also lead to a scenario where future generations lose certain innate human qualities or capabilities. The potential to engineer humans for specific environments might inadvertently lead to a reduction in genetic diversity, making the species more vulnerable to diseases or environmental changes. 5. Personalized medicine and genetic profiling. The report touches upon the concept of personalized medicine based on genetic makeup, which is indeed a significant advancement. However, the implications of genetic profiling extend beyond medical applications. If gene editing becomes commonplace, issues of genetic privacy and discrimination arise. Employers or insurers might seek to access genetic information to make decisions about hiring or coverage, leading to ethical dilemmas surrounding autonomy and consent. The potential for misuse of genetic information, including stigmatization or exclusion based on perceived genetic shortcomings, is a significant concern that deserves more attention. 6. Public perception and acceptance. While scientific advancements in gene editing are progressing rapidly, public perception often lags behind. The report emphasizes the need for societal agreement on the use of such technologies, but it does not delve deeply into how public opinion is shaped and the factors that contribute to acceptance or rejection of genetic engineering. The fears surrounding the alteration of the human species are rooted in historical precedents, cultural narratives, and ethical considerations. Addressing these concerns through education and transparent dialogue is essential for fostering an environment in which gene editing can be responsibly harnessed for the benefit of humanity. Conclusion. In summary, the report on DNA editing and its implications for human adaptation to extreme environments presents a wealth of information that encompasses not only scientific possibilities but also significant ethical, social, and evolutionary questions. The potential to enhance human resilience, the implications of cross-species genetic transfer, and the societal impacts of genetic modification are critical considerations that warrant deeper investigation and public discourse. As we stand on the brink of this new frontier in biotechnology, it is essential to navigate these challenges thoughtfully and collaboratively, ensuring that advancements serve the greater good and respect the complexity of human life. 1. Applications of gene editing for survival in extreme environments. The potential for gene editing to enable human adaptation to extreme conditions is significant. Technologies like CRISPR-Cas9 allow scientists to modify human genes, enhancing resilience against factors such as high radiation, extreme temperatures, and low oxygen levels. By integrating genes from resilient organisms, e.g., tardigrades, humans could theoretically develop greater tolerance to environmental stresses, potentially allowing for sustained life in habitats ranging from high altitudes to underwater environments and even low oxygen planets. 2. Enhancing biological functions. Beyond mere survival, gene editing could fundamentally alter human biology. Enhancements could include improved detoxification pathways to tolerate toxic substances, increased metabolic efficiency to withstand temperature extremes, and even the introduction of photosynthesis-like capabilities to generate energy from sunlight. These modifications might not only allow humans to thrive in resource-scarce environments but could also revolutionize our understanding of human physiology and longevity. 3. Ethical and policy considerations. The rapid advancement of gene editing technologies raises significant ethical questions and policy challenges. Variations in national regulations create a complex landscape where some countries may embrace aggressive gene editing research while others remain cautious. The case of He Jiankui highlights the ethical dilemmas surrounding human genetic modification, showcasing the potential for both scientific advancement and ethical missteps. As gene editing progresses, society must navigate concerns about altering the human species fundamentally, ensuring that ethical considerations keep pace with technological innovations. 4. Interstellar exploration and genetic engineering. The ambition of interplanetary colonization necessitates a re-evaluation of human capabilities. Genetic engineering may become essential for overcoming the harsh realities of space travel, including radiation exposure and the physiological challenges of living in microgravity. The discourse surrounding human settlements on Mars and beyond underscores the critical role that gene editing may play in ensuring the survival of humans in extraterrestrial environments. This highlights the intersection of science fiction and scientific reality, where gene editing could be key to humanity's future as an interstellar species. 1. The case of the He Jiankui gene-edited babies 2018. In November 2018, Chinese scientist He Jiankui announced the birth of the world's first gene-edited babies, twin girls named Lulu and Nana, who were modified to be resistant to HIV. He used CRISPR-Cas9 technology to alter the CCR5 gene, which encodes a protein that HIV uses to enter human cells. This groundbreaking experiment ignited a global ethical debate regarding gene editing in humans, particularly concerning the long-term implications and potential unintended consequences of altering the human germline. The backlash was significant, leading to He Jiankui's imprisonment for violating ethical standards and regulations on human experimentation. 2. The Human Genome Project 1990–2003. The Human Genome Project was a landmark scientific endeavor that aimed to map and understand all the genes of the human species. This international research project was initiated in 1990 and concluded in 2003, yielding a complete sequence of the human genome. The project has had profound implications for genetics, medicine, and biotechnology, paving the way for advancements in personalized medicine, gene therapy, and genetic engineering. The knowledge gained from the Human Genome Project is essential for contemporary discussions on gene editing, as it has provided insights into genetic variations and their relationship to health and disease. 3. CRISPR-Cas9 technology development 2012. In 2012, biochemists Jennifer Doudna and Emmanuel Charpentier developed the CRISPR-Cas9 gene editing technology, which revolutionized genetic modification due to its precision, efficiency, and relative ease of use. This breakthrough made it possible for scientists to edit genes in a wide array of organisms, including plants, animals, and humans. The technology has been used in various applications, from agricultural improvements to potential therapies for genetic disorders. The rapid advancement of CRISPR has raised ethical concerns about its use, particularly regarding human germline editing and the prospect of designer babies, echoing many of the debates ignited by He Jiankui's work. Future outlook: humans living in extreme environments through DNA editing. As we look ahead to the future, the intersection of genetic engineering and human adaptation to extreme environments presents a remarkable potential for humanity. With advancements in DNA editing technologies, particularly CRISPR and its successors, scientists envision a world where humans could thrive in high-altitude regions, underwater habitats, or even on low-oxygen planets. This future is not merely speculative, it is grounded in ongoing research and technological development. 1. Enhanced adaptation to extreme environments. The capacity to survive in extreme environments hinges on our ability to modify human physiology. Genetic editing can enable humans to endure conditions that are currently inhospitable. For instance, high altitudes. At high elevations, the reduced oxygen levels can lead to altitude sickness. By editing genes associated with oxygen transport and utilization, humans could develop enhanced hemoglobin levels or increased red blood cell production, improving their ability to thrive in such conditions. Underwater habitats. Living underwater poses challenges such as pressure and the need for efficient oxygen extraction from water. Genetic modifications might allow humans to adapt features similar to aquatic mammals, such as enhanced lung capacity or even gills, facilitating a more comfortable existence in submerged environments. Low-oxygen planets. The potential colonization of planets like Mars requires significant adaptations. Genetic modifications could involve altering metabolic pathways to optimize oxygen usage and resilience to lower atmospheric pressures. 2. Biological resilience and health improvements. Gene editing goes beyond mere survival, it can enhance overall health and resilience against environmental stressors. Radiation resistance. As space exploration becomes more ambitious, astronauts will face increased levels of cosmic radiation. By integrating genes from organisms that exhibit radiation resistance, such as tardigrades, humans could potentially enhance their cellular defenses against radiation damage. Reducing cancer risks. Detoxification capabilities. Genetic modifications could improve the human body's ability to detoxify harmful substances. This may involve introducing genes that bolster liver functions or enhance cellular resistance mechanisms, enabling humans to thrive in environments with heavy metals or pollutants. Thermal tolerance. Extreme temperatures, whether hot or cold, can pose significant threats. Gene editing could enhance the expression of heat shock proteins and other metabolic regulators that improve resilience to temperature extremes, allowing humans to adapt to varying climates across different planets or environments. 3. Ethical and policy considerations. While the scientific potential is vast, the ethical implications of human genetic modification are profound. The discourse surrounding gene editing is rife with concerns about the long-term effects on human evolution, the possibility of unintended consequences, and the moral implications of altering the human genome. Global policy divergence. Countries vary widely in their regulatory approaches to gene editing. Some nations like Germany and Canada enforce strict regulations, while others such as China have shown a willingness to push the boundaries of genetic research. This disparity could lead to a race in genetic advancements, with countries that embrace gene editing for adaptation potentially gaining significant advantages in space exploration and colonization. Public perception and opposition. The modification of human DNA raises public fears of playing God and altering the essence of humanity. Engaging with these concerns through transparent discussions and ethical frameworks will be crucial as we navigate the implications of this technology. 4. Technological breakthroughs and future research directions. The research landscape is rapidly evolving, with breakthroughs in gene editing techniques paving the way for more precise and effective modifications. CRISPR and beyond. The advent of CRISPR-Cas9 revolutionized genetic editing, making it accessible and cost-effective. The development of base and prime editing techniques allows for even more precise alterations at the genetic level, which could enhance the safety and efficacy of proposed modifications. Interdisciplinary research. Future advancements will likely stem from collaborations across genetics, bioengineering, environmental science, and space exploration. Understanding how various organisms survive extreme conditions can inform genetic strategies for human adaptation. Personalized medicine. As gene editing technologies mature, the prospect of personalized genetic modifications tailored to individual needs may become a reality, enhancing not just survival but also health outcomes for astronauts and colonizers of new worlds. Conclusion. The future of human survival in extreme environments facilitated by DNA editing holds exciting possibilities. As we continue to explore the frontiers of space and our own planet, the ability to adapt biologically will be paramount. However, this journey must be balanced with ethical considerations and societal consensus to ensure that our advancements serve humanity positively and responsibly. The potential to redefine human existence in extreme conditions is within our reach, but how we navigate this path will shape the legacy of our species in the cosmos.